Okay, so I think we'll go ahead and get started. So the tutorial that I'm going through, the files are under a folder called DIA on your USB drive. So go ahead and start copying those over. I think it's a few gigabytes, so depending on your computer, it might take a little bit to copy. Um, I'm actually just going to talk for a little bit before we actually start. So you guys don't need to follow steps or anything for this. So I'll just kind of talk as you copy stuff. Um, So talking about sort of the DIA data analysis workflow that I mentioned before, uh, typically we do hypothesis generation, we choose peptides, extract chromatographs for them, integrate the peaks, and do transition refinement. The first step though is, remember I was talking about how do we know even what peptides do we want to query from the DIA data? That's the first thing we have to decide is what peptides do we interrogate it for. Some things you can do is, for example, if you're interested in a particular pathway, you might take all of the proteins that are annotated for that pathway, do an in silico digest of them, and you say, I want to query the data for all of those. You could have a particular complex you're interested in. You might just want to go for a Go term, or you might even have particular modification sites that you want to extract peptides for. Every time I tell people this, they just say, well, I want to query for everything. And I say, great. There's a problem with that though, and that's when you query for everything. I generally don't recommend it. It for every single one of those queries, that's a hypothesis test. So if we're trying to make detections, you're testing a hypothesis. And the reality is, if you just take a FASTA file and you digest it, and then you look for every single one of those peptides, we know based on how mass spectrometry works that a lot of those peptides might not ionize well, they might not be detectable, they might just have a ton of interference, so you won't see them. So you kind of want to narrow your list down. But I know that a lot of times when you're digging into a DIA data set, you might not really have a hypothesis yet. It's hypothesis generating data, so where do we even start? And that's what I'm going to show in this particular example. We're just going to start with peptides identified in a DDA experiment. This is a really popular workflow, especially when DIA first is getting started. You would just do a few DDA runs prior to your DIA analysis, and then map those peptides over to DIA. So that's taking all of my comprehensive stuff and everything I said about why you wouldn't want to do DDA and kind of throwing it out the window. You are limiting yourself to peptides you identified in DDA in this case. However, you're using the DIA data to have better quantification on those peptides because of the better selectivity. So this is an easy sort of starting method for people to get their heads around. It's not what we actually do in the lab, but that is what we would do. So imagine that we have these samples and we just did, say, 3 DDA runs in the beginning and database searched them. That's what I'm going to take you through and how to do DIA-based quant for those. Later on, we're going to have a presentation where you would have generated a library where you do, say, chemical fractionation and then DDA to get a deeper library and analyze those in DIA. And later on, even further in the course, Brian will talk about generating a chromatogram library. But for now, just imagine we've done some DDA runs and we're going to query the data. So I want to just show a demonstration of actually what happens. So don't actually follow along with this. I'm just going to show you on the screen. But I think it's nice I kind of want to prove to you guys what I said about just trying to extract all of the peptides. So what I'm going to do is I'm going to take some DIA data. I'm just going to take 2 proteins, and I'm just going to extract all the peptides from it in Skyline. And I'm going to show you guys what that looks like, just so you know why I think it's a bad idea. Sorry, let me just get organized. Okay, so starting from a base Skyline document, again, you guys don't need to follow the steps. You're going to be doing these steps later in the tutorial again, but it's nice to just watch it rather than frantically clicking, at least for a first pass. So, I'm just going to make a new Skyline document, and the first thing is transition settings. So you guys by now are fairly familiar with this. This is a DIA acquisition method, obviously. These particular data were analyzed on an Orbitrap. The data were 17,500 at 200 m/z. So I think you guys are probably pretty familiar with this sort of thing. Resolving power here is 35,000 at 200 m over z. In this case, I'm going to include all matching scans. So this is important, right? So this is one problem of trying to analyze DIA data with no prior information whatsoever, is we're going to have to pull out every single MS-MS scan for every single peptide. So we're going to have these full chromatograms, and you'll see why it's challenging, because you'll have no idea where the correct peak is. If we have a DDA library, we can click this and say narrow it down within 2 minutes of where we identified it. So there's a lot more selectivity if you have a library to work from. But for now, I'll just say that I'm going to include all of the matching scans. Yeah. So in this particular case, I'm not using centroiding because this is sort of before this tutorial was designed before that was available. But yeah, that's right. Yeah. Yeah. So anyway, where was I? So then the next thing for DA is you got to tell it, Skyline, how the stuff was isolated. So in this case, I'm going to show how we would create an isolation scheme. So again, you don't need to follow this, but you set pre-specified isolation windows, calculate start MZ 400 to 800. So this is the range that the DIA method is going to cover. Then you can set the isolation window width. In this case, we'll do 20 MZ, and optimize window placement, which will try to set the edges of the windows in places where peptides don't occur. And then I'll just hit OK. And now Skyline has already generated telling us where we should place the MS-MS isolation windows. So this is a list of start and finish. You can kind of see that these windows don't have like integer values, and that's because those isolation edges have been optimized. If you click graph, you can actually see what the scan cycle looks like. Okay. So now we've kind of told Skyline, well, Skyline's told us this is how we're going to acquire the data, and then I'll just call this 20 MZ DIA. Hit OK. All right. Then the next thing that's important for DIA is setting this transition settings, the filter. So we'll tell Skyline, okay, how do we want to extract the data? So for any given peptide that it pulls from the FASTA, we'll tell it that we want precursor charges 2 and 3, and then ion types. So we'll have it pull out Y ions, B ions, and precursor ions. And then we'll do ion 3 to the last ion for each of those. All right. And then making sure that in the precursor settings, 1 second, so hit OK. What's that? Oh, okay, so I'll uncheck that. Okay. And then settings, peptide settings. Where was I? Modifications. So I just need to make sure that this is set correctly. So these are carbamidomethylated peptides because they've been treated with iodoacetamide, and that looks correct. Okay. And then in the peptide settings, I'm going to say make sure that we have no library selected, because again, we're just doing this in the naive approach. I just have a DIA data set. I'm just going to try to pull out information for proteins. Oh, do I? Oh, I see. No, I don't have any heavy labeled peptides in this particular example. This is just completely just label free. Okay. All right, so in this case, I've already prepared. I think you guys have already covered background proteomes. Oh, there's a yeast proteome right there. Maybe I'll just use that one. Oh, well. Yeah, I can do, well, no, I'll use mine. Let's see. Should already have one. Oh, I see. I need to generate one. Okay, I'll just use Brennan's because it'll be faster. Okay. So now what I should be able to do is just insert proteins from yeast by clicking here. And then I'll just insert pyruvate decarboxylase. Okay. And then I'll insert okay, and this is generated just like a list of peptides from doing an in silico digest for these particular proteins, and then I'll turn around and import the data. But I'm going to take a shortcut because I actually have a Skyline document with it imported. Oh, because they're outside of 400 to 800 M over Z? Oh, was I not supposed to answer that? Yeah, so in the settings, transition settings, right, so remember when I defined the isolation scheme, I told it Skyline has knowledge of where it expects the windows to be. So it's just kicking out any peptide if the precursor is outside of that M over Z range. Okay, so then if I open the actual file where I've imported this, so here's an example of what things look like imported. So I just want to show everybody. This is what DIA data would look like if you just tried to pull out every single peptide. I'm going to make it so we can see the precursors. Oh, I guess I don't. Oh, right. Okay. Are the colors all different? This is colorblind mode? Oh, okay. So this is just showing for this particular peptide, right? So we can see that there's a signal for it. But if you were going through and manually doing this, wow, that was way over zoomed. Sorry. So you can imagine with no prior information, I might be able to show Mike McCost this peptide and he might believe me when he was my PI until maybe I zoomed out. And then he might say, well, I'm not quite sure that's the peptide. You know, there's all of these other options. You would end up clicking on another peptide, and there's the legend's too big. And now you'd run into this problem of you can imagine yourself trying to parse through this data and looking at the peptide and looking for every peak and saying, well, I guess that's the best peak for the peptide, but I'm really not sure if that's the one. So that's the problem you run into trying to just analyze DIA data like you might SRM. One way to get around it would be to have heavy labeled standards, so you can then know that that's where I expect it to be. If it's within the retention time colluding with the heavy standard, then the peptide's there. But you can only then do quant on the things you have heavy standards for. So the way we need to get around this problem is to have some sort of algorithm for detecting the peptides directly from the DIA data, or in the case that I'm going to show, mapping detections from a different run, so from a DDA run into the DIA data. Okay, so some of the problems we run into of trying to analyze the DIA data without any prior information is the presence or absence of a peptide peak, selecting the right peak, and then setting the integration boundaries correctly. So now we're going to imagine that we've done an experiment like so, where DDA data was acquired alongside the DIA data. So imagine that you had a run here with DDA and a run here with DDA, and then some DIA in the middle. This is still kind of a limited analysis, but it's good for demonstration. You're limited to only these DDA ID'd peptides, which is definitely not optimal. But it's a good way to get a jump start and just to get your feet wet with DIA. Okay, so now we'll jump into it. So now I want people to start following along as we click through this. So everybody should have successfully transferred the DIA folder from the USB stick onto your hard drive. Please make sure you're working off the hard drive, or else this will really be slow. All right, I'll close out of here. Whoops. All right, so for now, we'll start with this blank document. Presumably, everybody's with me at this point. So click on Settings and then Transition Settings. So the first thing we need to do is explain to Skyline how the DIA data were acquired. So mine's already populated, but let's go ahead and do it in centroided mode. We'll just do it live today, and I will ignore my old settings, and we'll make sure, well, I'm sure everything will go smoothly. So, so Brendan pointed out, you guys will remember when I demonstrated to you at first that I clicked Product Mass Analyzer Orbitrap, and I put in the resolving power that I acquired the MSMS data at. So in this case, the DIA data is actually acquired with profile MSMS spectra and profile MS1 spectra. And that's what these settings in Skyline represent. But I'm actually going to have everybody switch it over to a centroid analysis. So what happens when you do that is Skyline will centroid the DIA MSMS data on the fly as it's being imported. And then the extraction windows turns into a tolerance on mass accuracy rather than resolving power. So now we're saying we expect the centroid of the peak to be within 10 parts per million of the theoretical mass to charge. So go ahead and set that for both the precursor and the product ion. We're going to have Skyline centroid the data and only extract centroids that fall within 10 ppm. That's what we do now. So when I actually acquire data, I do MS1 in profile and MS2 in centroid. It just becomes, the profile data becomes so large, the files are so big that and I don't see a big benefit to having the profile data over the centroided, so I tend to just go ahead and acquire centroid. That's speaking for the instruments I'm familiar with, so that's going to be the Fusion series and the Q Exactive series. Yeah. So the isolation scheme that we have for this particular data was acquired using just these 20 MZ windows. So we're going to set that in a minute. That's okay. But first, the retention time filtering. So before, I had no idea of where any peptide was supposed to elute. But now we know we're going to have some information of that. So we're only going to extract data for retention time around where the peptide came out in the DDA identification, in the DDA runs. So you can set this to use MSMS scans within 2 minutes of the IDs. And then for the isolation scheme, first let's start with just clicking add. So I want to remind you guys what I did before to actually generate the isolation method with Skyline. So if I set 20 MZ DIA as the name, and if you wanted Skyline to generate an isolation scheme for you, you click pre-specified isolation windows, hit calculate, and then for the start MZ, in this particular case, we did 400 to 800, but you could do 500 to 900, and set the window width to 20 M over Z. And then select the optimized window placement. Everybody good? Good. All right, so hit OK. And then you can graph the isolation scheme that Skyline created by clicking that graph button. Okay. So that's if you wanted it to generate, I'm going to tell him, Brennan, don't worry. So that's if you wanted to generate the isolation window for your instrument. However, if you then wanted to import data, you have this other option, which is to use results only. Is that what you wanted me to show them? Oh, it'll import from the data file? I've never used that feature. Pressure's on. Right.
That we made it possible for Scott to just figure it out. And figures out, using swap overlapping windows and all that.
Cool. So yeah, if you wanted to, so in this case, I'll do that a little more slowly. So we had, in this case, the raw files. Let's say we've already acquired them and we didn't necessarily know what the isolation windows were. If you wanted to tell Skyline explicitly what they are, you can click import, and then in this case, the raw files under DIA tutorial raw, and you can click on either one of these files. They both have the same isolation scheme, and click open. And now Skyline is going to read the isolation scheme from the data.
Which really could be a lifesaver if you're trying to get it from somebody else. Actually, you have some data from Google where they made a mistake in the isolation scheme and then the graph and all of that. The graph will show you.
But when you graph mine, you'll see there's no mistakes. That's the rounding. That's just thermo rounding the value we told them to. Yeah, 0.0001 is the gap, so that's a rounding error. That's not a mistake. Okay. So does everybody feel comfortable where we're at? Any questions? All right. Okay, so now we've hit okay. So now Skyline kind of understands how the data were acquired. It knows where all of the isolation windows are. Yeah, so you can switch this to results only also. And that's it's basically the same thing as telling Skyline to import the scheme. It just reads it from the file on import. So you should get the same results even if you have results only set or not. But I want to use I actually want to use the one that I defined before because it actually knows 400 to 800 M over Z.
Drop that again. Right below results only is also results only with 0.5 margin, which is the typical sort of science swap type.
Yeah.
So if your results only will work great if you have no overlap, no margin, like your data.
Yeah.
But if you have science data, you want to use the other.
Right.
Yeah, have one delta, and that's just like that's the standard, most often published science.
So, question, Brennan, if I just import with results only, so if I have this set to results only, and then I go to my DDA library and I want to add peptides from it, is it going to add stuff to the document that's outside of 400 to 800? Right. So then when I turned around to import, I would have a bunch of peptides that are outside the range, right? Okay, so I want to use this 20 MZ DIA because that we generated from the import of the file. The reason being because then it's telling Skyline a little bit of extra information, which is I only want to use peptides from 400 to 800 M over Z. Okay. Everybody good? All right. Okay, so now Skyline it understands how the data were acquired, but we need to start telling it how do we want it to query the data, right? So what transitions to pull from and stuff like that. So go ahead and click the filter tab. And mine's kind of already filled out from before, but precursor charges 2 and 3, that's just going to say for any given peptide, pull out the doubly and triply charged precursor. Set the ion charges to 1, so that's the fragment ion charge that's going to be pulled out. And then for ion types, Y, B, and P. So that's the Y ion fragment series, the B ion fragment series, and the precursor series. And go ahead and set product ions to ion 3 to last ion. And then we can unclick this N-terminal to proline thing. Brennan already explained what that is. And then for this use DIA precursor window for exclusion, I tend to not use that, but I guess some people use it because what that's doing is, well, has this feature been explained to you guys yet? Okay, so what use DIA precursor window for exclusion does is it says let's say I'm extracting a peptide from a window that's covering 500 to 520 MZ. That's the isolation, right? What this will do is it'll say, I'm not going to take any fragment ions that fall between 500 and 520 because that range of the fragment ion window tends to be a little bit busier. So you can have more noise in there. So some people like to exclude any fragment ions that fall within the isolation window itself. Okay. All right, so now I'm just going to hit OK in this window now that we've kind of defined what sort of transitions to extract. All right, so now the next step is we need to build a spectral library. So we've assumed now we've acquired some DDA data files and we've searched them. So we've got a bunch of raw files. We've done, I think in this case, it was like a sequest search. And we need to start and put together a library that Skyline so Skyline can interpret those search results. So if you hit settings, peptide settings, and click on library, and then click on build. All right, so we're going to put together a library for yeast. So let's call it, I guess don't put my name behind it, but say like DIA tutorial yeast, because apparently there's other yeast stuff too. And then for output path, I already made a folder for folks to put it in if you want to just mirror what I'm doing to make it simple, but feel free to put it wherever you want. Under DIA tutorial, I made this sort of spectral library folder. And you can just save it there. So I'm going to unclick this keep redundant library. That's what the reason for keeping a redundant library would be if you ever see in the future that you think you want to expand the library, add on more search results in the future, you need to have the redundant library existing to do that. For the cutoff score, I'm going to put 0.99. So in this case, the DDA data was searched using a combination of sequest and percolator. Percolator, the confidence value that it returns is a Q value. Roughly, it's similar to a false discovery rate measurement. And so by setting a cutoff score of 0.99, that corresponds to a Q value of 0.01. So we're going to basically accept peptides with an enforced false discovery rate of 1%. The reason the cutoff score is 0.99 and not 0.01 is you need some sort of normalized scale between all these various pipelines. And some of them a higher number is better, some of them is a lower. But generally, putting a higher number in this cutoff score means it's a more stringent cutoff. All right. So then click Next. Okay. And now to add our input files. So if you click Add Files, there's a folder again under my DIA thing. So it's DIA tutorial, Spectral Library. And this has the search results from my search. So I'm going to show you guys, I'm just going to click all files to show you the entire contents of the folder. These are CMS2 files, so these are files containing MSMS spectra. The SQT files are the direct sequest output, and all.perc.xml is the output from Percolator. So that all.perc.xml is the thing that has sort of the calculated confidence and statistics from Percolator. Okay, so now I will just hit the all.perc.xml and hit open. And then click finish. It should build the library. So it very quickly ripped through that library. So it looked at the percolator, said I'm going to pull out the peptides Q value 0.01 or less, and then I'm going to go to the CMS2 files and pull out the spectra. Some peptides spectra matching the following peptides were excluded. So I think that's when one spectrum maps to multiple peptides. Brennan, can you explain that one? I always forget. The spectra matching the following peptides and multiple ambiguous matches were excluded.
Yeah, so that's where they will assign spectra to multiple.
Right. Okay. Yeah.
Without any differentiating type of score.
Right.
So we just say like, well, those are both.
We don't know. Yeah.
We allow you to keep them. There's a checkbox to say you want to keep them, but we don't. We would recommend mostly that you keep them in the case of like DDA, where they're just going to be used replicating MS1 signal.
Yeah.
In a case where you can actually be using them
To select fragment ions.
to select fragments.
Yeah.
You don't really know what the fragment is.
Right. You don't know what they're from. All right. So then just go ahead and hit OK. Okay. So hopefully everybody now is at this point. So did anybody have any problems with the library building? Excellent. Okay. So just hit OK on peptide settings, and then we'll just make sure that everything worked. So if you hit View Spectral Libraries, you should be able to now see the library. So make sure you have DIA tutorial yeast selected up here. One thing that's really important when building these libraries, especially in this workflow, is you want to make sure that there is a retention time value displayed here. That's just indicating that Skyline successfully read the retention time for each peptide from the file. Okay, so now we have sort of a set of peptides. A set of peptides, but Skyline doesn't necessarily know the full protein sequences of all of the yeast proteins. So I'm going to click this. Do not click what I click. This is demonstration purposes only. But if I just hit add all, whatever, add all, sure, just add them. Then you'll see what happens, right? Sure, you have a list of peptides, but it's not as useful because they're not associated with any sort of proteins, right? So we need to tell Skyline, here's the FASTA file for yeast, so it knows how to map those peptides to proteins. So I'll remove this and undo that. Okay, and now we'll go ahead and generate that background proteome from a FASTA file. So if you go to Settings, Peptide Settings, and click on the Digestion tab, we're going to create a background proteome. It's going to be for yeast again, but let's use my FASTA file this time to make sure everything works. So just click Add for background proteome. Maybe name it like DIA tutorial yeast to help keep it straight. All right, and then we need to create a file. So this will create a protdb file, which is Skyline's own format for keeping track of background proteome sequences. I'm going to put it under DIA tutorial FASTA. And I'll just call it, I guess, yeast or DIA tutorial yeast. And hit save. All right, so now it's created an empty file with no sequences, and now we need to add FASTAs to it. So we could, if we wanted to, we could add multiple FASTAs, but I've got it in one. So again, browsing back to that FASTA folder. I have a yeast FASTA there that you can use. Sorry, I kind of clicked through that fast, but I think you guys know DIA tutorial FASTA. And then just find that yeast FASTA and hit open. Yep. I don't know, Brennan. What's the difference between doing the FASTA from a background proteome versus doing file import FASTA?
Well, so background proteome just sort of provides an environment of what's supposed to be in your sample, and import FASTA will create targets. So there's what you're targeting in the targets view versus the background is. So you could be targeting two proteins in a background of human blood, or in a background of yeast or chicken blood or whatever.
Oh yeah.
So the background proteome allows you to say things like, well, is this peptide duplicated in my background? So there's extra information that we can get from having a background proteome. But the background proteome doesn't affect your target list at all.
Yeah, like
I'll find unique peptides, yeah, so it's and that ability that Garrett showed where he just typed in a protein name.
Yeah, that's what I was going to do real quick.
That comes from the background proteome. So a lot of information can be taken from the background proteome, but when you do file import FASTA, that's a put everything in my targets list.
I also have a similar question. Is there a difference from doing all these steps from here than from the start menu?
Oh, the wizard? I mean, the results hopefully shouldn't be different, but I don't think the results should be different. It's just two different ways of going about it.
Well, yeah, everything you can achieve from the wizard, you can also achieve just doing it from here. It just turned out you were getting a sense that trying to teach everybody how to do everything all from settings was blocking people, and they were feeling like, where do I start? So that's why we created the wizard. There's nothing about the wizard that you can't achieve if you're an expert with that.
So go ahead and set the maximus cleavages to 2. Okay. And so actually I do have a quick demonstration. So I'll hit OK, and I'll just show you guys, I mean, what Brennan was just talking about. So with the background proteome set, right, that's how now when I start, remember I did this before, I just put in pyruvate decarboxylase. Whoops. This would be a particular example of, say, you're just interested in targeting it, not the whole proteome. So now the targets list is just a subset of the complete FASTA. Another thing to observe is in the prior document, where I had done with no library, you can see that the list for pyruvate decarboxylase is much longer. And that's because now Skyline's automatically filtered it down to only peptides that are in the spectral library. So, and that's what this symbol here indicates. That little spectrum thing means that it's pulled from the spectral library. Okay. So if we go to now the settings, transition settings library. We can tell Skyline kind of how we want it to pull down, how we want it to choose transitions from the library. So, for example, if I say the ion match tolerance of 0.1 m/z, that's saying how similar. So for any one of these fragment ion peaks, the theoretical fragment ion, Skyline's going to try to look in the library spectrum and find that fragment ion. And this is saying what's the tolerance for matching the two. So in this case, I'll set it to 0.1. And then for the if a library spectrum is available, we'll have Skyline pick the 5 product ions from the filtered ones. Or are they what? Cleaned? Oh, you mean like versus just opening it in the spectral library viewer?
It should be the raw spectrum. Yeah, if that actually corresponds to a spectrum that was provided us, there are certain tools that like MaxQuant, need to contact Jürgen about this, but like there are tools that will do things to spectrum, in which case they may not end up being exactly wrong. But our library building tool uses the spectrum that the search tools provide us.
It's not like maximum what we have.
So yeah, unless you're using, you can be using a library from X Hunter, which uses a maximum of like 20 peaks. And that, but Skyline's not doing that so much.
Okay, are people on the same page? Here's some mumbling. We're good? Okay. So yeah, actually it's a relevant point. I mean, when bringing up this spectrum viewer, you guys can sort of see this is the spectrum. Of course, there are some peaks that aren't part of the fragment ion series that we told Skyline to extract, right? So we're just looking at singly charged, or I guess multiply charged also, Y and B ion peaks. But some of them could be a part of a different ion series. I mean, I suppose you can look for all kinds of different ones, but we're only going to be extracting peptides that are the top 5 most intense out of the series that is in the filter window. So of the Y-ion series, for example. All right, so I'm going to close out of this. So everybody just make sure under settings, transition settings, under library, just make sure you've got this set to 0.1. If a library spectrum is available, pick its most intense ions and then set it to pick 5. Just make sure everybody's got that. Okay. And then you can see now that it's selected just for this particular peptide, right? It's picked for the top 5. We typically find that, it's, I suppose you could want to look for doubly charged product ions for sort of more highly charged precursors. In this case, we're limiting the analysis to doubly and triply. I think if you're going for something higher charged, you could then model those doubly. But the more sort of fragment ions that you try to model, the more chances you have of kind of choosing a crummy one. So it's kind of, if you don't expect there to be a lot of doubly charged fragments that are good for quantitation, then you wouldn't want to include them in your query because you might pull out one with interference. So one of the limitations of the sort of using DDA spectra for picking the fragment ions rather than using, say, a chromatogram library is we may have a really high fragment ion that has a really good intensity. That doesn't mean it's not going to have interference. Right, it could just be a heavy interference and we wouldn't know because we don't have any chromatographic information. So I try to avoid including, I try to be a little bit more stringent rather than opening it up and just letting anything in. Okay, so now that we have the library, I think one thing I wanted to show people is if we try to insert a protein by name, so I guess, well, you guys might not have the thing up, but get to this box where you can insert a protein by name. And then if I try to insert, remember before I was talking about TOR1. So now I'm the grad student trying to study TOR1 and I hit enter. And what just happened? Yeah, why? What's that? That could be part of it, sure, yeah. Yeah, it could have just not been identified in the library, right? So sometimes you'll add a protein and you'll find that it has no peptides because it's not in the spectral library. Anyway, so at this point, I want everybody to clear out the targets list because we're just going to insert everything from the spectral library. So I just hit Control A and then delete, but you can use any approach you want to do that. All right, and then click on this View Spectral Libraries. So this is our library view from before. And remember at first I just clicked Add All, and I just ended up with like a list of peptides. Now you can actually click Associate Proteins, and then hit Add All, and you'll see the difference. So it's now matching the peptides to those proteins. So now you'll have a proper target list hierarchy with all of the different peptides associated with proteins. So now we're finding that Skyline's informing us about some issues. One is that there's 266 peptides that are matching to multiple proteins. Do you guys think we should include those or not? How come? We'll have more proteins that way if we include them. Yeah. Yeah. You can't use them for quantification because you don't know if they're mapping to something else. Although I would be careful because what if your FASTA file contains multiple isoforms? What if it's one of those huge things with lots of isoforms? So I think there are ways around that in Skyline. So in the sort of enforced peptide uniqueness, which I kind of assume you guys will learn about if you haven't already, there are ways to get around that issue. But just keep in mind, just think about what you're doing with this. Generally, you don't want peptides that are going to be redundant amongst multiple different proteins. So I'm going to let's hit do not add for that. And then 21 of them don't match the current filter settings. So my guess is those are ones that are really short or really long. So we'll go ahead and hit do not add for those also. Oh, right, yeah, sure. If the database search included like 4 miscleavages or something, and then here we have it set to 2, yeah. So we'll hit do not add. I'm curious, does everybody have the same numbers, like 266 and 21? Okay, so some no, most yes. That's pretty good. Normally it's like 50-50. So just hit OK even if you have different numbers. The reason that some people's numbers might be subtly different is that some of the settings like propagate over from other changes or previous tutorials. So some of your settings might be slightly different. But go ahead and hit add all. And now we should have, yep, so now we have this fully populated targets list. This top protein is actually a contaminant. So go ahead and click that and delete it. You guys know I don't, see now you know I don't sugarcoat it for these sorts of projects. This is actually really bad data that we're presenting, but it makes, well, no, but it makes the whole analysis easier because there's way less peptides and proteins, so everything goes faster. It's just a tricky way of making everything move more quickly. Okay, so then I want to find a particular peptide. I'm just going to do it really quickly because I changed some of the settings. Okay, so I guess if you want to follow along, the peptide I just searched for, I hit Control F, and then I searched for a peptide SDVIFPILIPT. If you didn't get that, that's too bad. I'm not going to repeat it. Just hit find next. You can watch on my screen. So if I expand it on the screen, we can see that this peptide only has 2 transitions. I think you guys could probably tell why. Looking at this library match, we can see that, one, it's a very sparse spectrum. The spectrum did pass confidence in Percolator. I mean, we set it to roughly a 1% false discovery. Perhaps this is a false discovery. But you can see that Skyline only has 2 transitions for this peptide because there's only a couple that pass the filter. Why wasn't this Y2 included? What setting caused that to not happen? Yeah, the ion filter. So we told it only ion 3 to last ion. That's on purpose because this Y2 is most likely has interference in it. I like to filter out these types of peptides that only have like 2 transitions, and it's just a good thing to show you guys whether or not you want to do it. So to remove peptides like this, you can hit Edit, Refine, and then click on Advanced. And then we can set the minimum transitions per precursor. So in this case, I want to keep all precursors that have at least 3 fragment ions. So I'm going to put 6 there because the precursor, the M plus 1, M plus 2, so the MS1 peaks also count towards this. So I know I always have 3 of those, and then I want to have at least 3 transitions. So summing that up, you get to 6. And then auto select all precursors. So when you do this, it should then remove all precursors that have less than 3 transitions. So before I click this box, I'm looking down here at the number of precursors I have in the document, and that's 867. So now if I hit OK, it drops down to 848. So there's a few that didn't have those precursors, and now that peptide that I searched for before is no longer present in the document. Okay, is everybody on the same page here? I think I'm going to go ahead and import the data. So I guess let's save the document. It's probably a good idea. So click DIA. I think I have a place for it under DIA tutorial skydocs. Just call it DIA tutorial.sky. Okay. And then go ahead and import the data. So we're going to hit File, Import, Results. Add the single injection replicates and files because each file is a single injection. And hit OK here. Oh, well, I guess I should make sure everybody's box says the same thing. So make sure you have add single injection replicates clicked. Make sure this drop down says none. Under files to import simultaneously, go ahead and set that to many, and then select OK. And now we'll go to the raw files. So that's under DIA tutorial, raw, and just go ahead and select both of them. Click open. I'll just hit do not remove common prefix. And now the results should start importing. There they go. Does anybody have any questions while the data imports? No, I did not. So in this particular example, I'm doing it without IRT. Usually you would want some sort of retention time calibration, particularly if the library data was acquired either from an aggregate of experiments or on a different column. This is just the very simplest case where actually I acquired the DDA data on the same LC column. So we're just using like a 2-minute alignment without IRT. Oh, okay. I'll take you through how to fix that in a second. So if you're getting an IRT standard warning, just make sure you don't have an IRT calculator set under settings. Oh shoot, where is it again? Under settings, peptide settings, prediction. Make sure you have your retention time predictor set to 9. Presumably some people still are importing, right? Oh no, is it? Oh, why isn't this plugged in? Thank you. There we go. Oh, good looking out. Yeah. When it went up? Did it have heavy labels included? Let me see. That sounds like a dump, which makes me think that it's included heavy. If you just look at the templates. Yeah, you bet. So good as multiple people have it? Alright. Sorry guys, I should have had you clear this setting out. So some people are seeing that their number of precursors is on the order of 1600. That's because you might have heavy labeled peptides set from the previous tutorial. So if you have that, there's no worries. We just unclick it. So settings, peptide settings. Under modifications, make sure you have none of these isotope modifications clicked. Furthermore, it's probably a good idea to check your structural modifications and make sure that you only have carbon monomethylated cysteine selected. Good? Does that solve the problem? And hit OK. I mean, I guess you could have seen if there were some heavy labeled peptides in the data set, but probably not. Hopefully not. As someone who's used Skyline for maybe 8 years, this whole high contrast mode is really throwing me for a loop. Okay, are we okay to move on? Are people still importing? Okay, we're not really pushed for time, so if people have questions or anything, just ask. All right. Yeah, sure. Yeah, no, let's do that. So let's go ahead and I'll show you guys kind of how this is like kind of, I guess, everybody does this stuff differently, but this is how I usually set up my skyline window, so I can show you that. I'll close this for now. Usually I don't look at the library match, so I'll close that. But I usually do something like this. So I hit this like view retention times, and then a replicate comparison is the one that's going to give you the plot that I just had. And then I usually dock that somewhere. Oh, does it look like a single red block? Okay. Yeah. Yeah, if you click on a protein, it shows you all the peptides. This is really interesting. So people are having this view retention times graph, and they're not seeing this. And we're like kind of trying to figure out why. Okay, well, I think I'm just going to move along regardless of slight differences in the retention graph because I don't know why that's happening. So I'm unfortunately not of much help. So go ahead and dock the retention times graph. So if you click on, and I guess we'll do the peak area graph. So if you hit view peak areas replicate comparison, that's going to show you all the peak areas. So this is showing also the library spectrum. So you can see that we have very high similarity for this particular peptide between the actual peptides and the library spectrum, which is of course something that we want to see. In this case, the dot product, which is a spectral contrast angle, is 0.99 between the two. So do people see a plot that looks like this? If you hit view peak areas, you can't get the library. If you wanted to show you the library, you can right click and hit, where is it? I know there's a thing to do library. Oh, show expected. That'll show you the library. Do you guys have transitions all selected?
Yes.
And you don't have show expected? Okay, let me see one that doesn't have it. Okay, sorry. So to be able to see the expected, you need to split the graph out. So if you hit transitions, make sure you have all selected, and also hit split graph. So what split graph does is it shows you MS1 data on the top and MS2 on the bottom. What's that? Okay. Yes, okay, success. Okay. So hopefully now we have the peak areas graph set up, and we'll just dock that on the bottom. In this case, the legend's making it not show, so I'll turn off the legend. Okay, so now if you just click on any peptide, it doesn't matter which one it is. One thing that you usually want is, if you're not seeing the mass error, you definitely want to be able to look at that. So I'm right-clicking here and clicking mass error. So that's showing, for the DIA data, it's showing the difference between the measured M/Z and the theoretical. And you can actually plot any one of those. So if you mouse over one of these transitions and click on it, it'll show you the actual MSMS spectrum. And you can zoom in on it. So you can see that it's a really narrow 10 ppm tolerance and that it's off by just a little bit away from theoretical. And you can also zoom out by clicking the zoom to selection. So you can see with DIA that this is the DIA spectrum, not the library, right? So it's a very busy spectrum. In fact, it's kind of cool to, if you pull up the library match also and hit view library match, you can actually kind of see the difference between the two spectra. So on the right, this is the DDA spectrum, and then on the left, this is the DIA spectrum. So if you kind of click through some of these, looking, for example, on this peptide, we can put the library match up here and start to click on it, and you can see the difference between the DIA spectra and the DDA spectrum. Okay.
So if I'm seeing only the MS1 in the main window and just click graph to make it work,
Yeah.
it's not working.
To show it in this one.
So the MS1 is the same window.
You mean with it not split like this? But when you hit split, it doesn't actually split to anything? Yeah.
Well, it's not doing it on the key area. The mark is staying on
The legends library. That's just it's just because there's not enough screen, I would say. So just like that. Yeah. Okay. Do people sort of have any other questions about like visualizing the data? I know some people had trouble making the plots work and everything. Okay. So another thing for sort of setting up your view is if you hit view arrange graphs tiled, that will sort of try to arrange your graphs, and it doesn't work well if you have a lot of stuff open. But usually I end up with something like this. One additional piece of information that you definitely want to look at, or is helpful to look at when you're using a library, is you want to be able to see kind of where was the retention time in the library itself. So if you right click here, you can show, oh man, where was it? You right click and hit peptide ID times, and you can hit this from other runs. And now it shows this subtle little blue line for where the identification maps to, right? So Skyline imported plus or minus 2 around this. Wow, it's really hard to see, isn't it? There's a blue line there, I promise. Like, it's right there. Oh, no matter how much I zoom in. Yeah, good. Okay, so hopefully people see the blue line. And sometimes you'll find, sometimes you might be able to find sort of suspect detections where the blue line might be further away from the peptide than you think. If you're seeing that happen, one thing to keep in mind is if it's a peptide with a very low retention time, sometimes that region's more variable in terms of the retention time, so your expectation might be that it's a little further away. But generally, you want it to be pretty close. A lot of times with DIA data, you need to do manual refinement of transitions. So one thing I'm going to do is hit auto, is have the graph automatically zoom in on the peak. So if you right click on the graph and hit auto zoom x-axis and then select best peak, you're now zoomed in. And you can click through some of the peptides and actually find that the transitions seem to be pretty well refined. But one thing that I used to do is show people a peptide that has a bad example. But I'm pretty sure, oh good, it's still bad. Okay, so the peptide I searched for, if you want to search for it, is hit Control F. And the sequence is SNLAGK-PVIC. Good? Okay, so looking at this peptide, it looks like we got a transition that split through the cracks, right? So what happened is this transition in the library spectrum, no doubt, is one of the top 5 most intense. But from that library, we still don't, that doesn't mean that none of the transitions are going to have any interference. So looking at this particular transition, what you can do is actually try to figure out what it is if you actually show the raw spectrum. So I'm actually going to click with this circle over the transition and go look at the spectrum. And then zoom in on the selection. What we're seeing is that this is this Y7 thing. That's the theoretical monoisotopic mass for the fragment we're trying to pull out. But clearly this is an interference because whatever this is, it's not the monoisotopic mass of the fragment. This is some other fragment where we're hitting the m+1. So in this particular case, we would want to remove it. So we can just find the transition in the targets list. It's this one, and hit delete. That's one way to solve the problem. Of course, then you're manually going through every single peptide and choosing transitions, which you could do, but I think it's one benefit of sort of using, for now, what Brian's going to present, his encyclopedia algorithm later on does a good job of automatically removing transitions with interference. If I had left this transition in, another thing actually I wanted to show people is looking at this MS1. You guys see anything weird about this MS1 up here? Yeah. Yeah, this right here, right? Okay, so I want people to try to figure out what's happening. Does anybody have like a hypothesis of why, of what could cause that? Yeah, who said that? Mass error. So what's happening is, go ahead and click on this. What color is this? Orange? I'm colorblind, so this totally screws me. But whatever color this thing is, click on it. And it should load the spectrum. Okay. And then go ahead and zoom in on the particular peak. So this is now showing the monoisotopic, the m+1, and the m+2 for this spectrum, okay? And then I'm going to zoom in a little bit further onto just the problematic peak. So we can see right here that the peak's right inside of the integration boundaries, the 10 ppm boundary. But now you can walk along by spectra. So if I click this right arrow, it's going to move along. And see what happened there, where it drops out, that's a mass error issue. So this is one challenge with processing centroided data. The data's been centroided and we set it up for a 10 ppm error tolerance. If we had looked at the profile for this data, it could be that there's some interference arising here, and the interference is causing a little shoulder that drifted the centroid outside of the 10 ppm tolerance window. So some people will actually, when they analyze centroided data, they might do a higher tolerance, like 30 or 50 ppm, to avoid cases like this. What I can tell you is if you analyze the data in profile mode, so you said extract with resolving power 17,500, you'll have less of those cases where it drifts out, but you'll also have more interference. So it's kind of a trade-off. But this is just to demonstrate, how this sort of visualization of the data can be really, really helpful with determining why problems like this arise, and you might determine that you want to then re-import the data with 20 ppm to avoid this. Okay, now I want to jump to another peptide. I'm going to jump to it first to make sure it's in here. Okay. Okay, so if you jump to the peptide is TYAA EIA HNISAK. Okay, so we can see that this particular transition Y11 has a red dot next to it. It's really important. This is like a total gotcha. So I think you guys may have already heard this, but when you're doing these sorts of things in Skyline, you definitely want integrate all to be selected. It's like one of the first things I look at when I'm setting up a document is I make sure settings integrate all is chosen. So if I unclick integrate all, see what happened in this plot over here? I'll click it again. So if I go back to integrate all, it's got all of the transitions and it's integrating every transition that it shows me. But if I unclick integrate all, it only will integrate the transitions that it thinks are clean. The problem is sometimes the transition will only be clean in one run, but it'll be fine in another. So you'll be essentially integrating different transitions in different runs, which is really going to screw up your quant. So you always want integrate all to be selected. Yeah. Yeah. Oh, this one. Okay. So what it shows you in the full scan window is based on the last thing you clicked in one of these windows. So let me make this a little bigger for you to see. I'm going to bring this guy back up. Where'd he go? Okay. So it's based on what you last clicked. So to bring up the full scan window in the first place, you kind of click on a transition and it plots it for you. So right now mine's showing MS2 because that's what I clicked on. And then to switch it back to MS1, I just click up in the MS1 data. You can also click this drop down here. Oh, I guess you can't. Never mind. I thought you could. Yeah. Yeah, and then you can walk along the scans of this. But yeah, I guess you have to just like click in the MS2 or MS1 data in the plot. Any other questions? Good. Okay. So one other important thing for this type of DIA data is I've already told you guys that when we do the quantitation, we only want to do the quant with the fragment ion data, not with the total integrated area. So I'm going to show you guys a few ways to drill in on that. Yeah, so I'm going to show you guys. Let me show you really quick. So if you hit View Results Grid, have you guys been introduced to the results grid yet? Yeah. Okay. So if you click the results grid view for a particular precursor, you can see some of the summary statistics that Skyline has calculated. I think one major gotcha is I know a lot of people, you'll have Skyline data and then you'll export it to a report or a spreadsheet. And if you're doing this DIA data where we've imported both fragment ion and precursor, and then you just give it the total area, that's actually the summation of the fragments and the precursors together. So it's adding both of those. That's not what we want to be using for quant. For quant from DIA data, we want only the integrated signal of the fragment ions. You mean the summation of both? I can't think of any. But keep in mind that when Skyline was first made, it was only SRM, so you would almost never have like any MS1. So it was just total area, and I think that the saying total fragment area was added after the fact. So in order to do the total area in the results grid, the way to do that is if you edit the view. So if you hit View, Customize View, you can select what columns are shown. So for the, and you can see here under precursor result, you've got the total area, so that's the sum of everything. But you may want just the total area fragment. So if you click this box for total area fragment and hit OK, oh, the view name cannot be blank. There we go. Now it's going to have the total area fragment displayed. And that's the number that you want to use for quant from DIA. You don't have, none of you guys have those options? Are you clicked on a precursor? Sorry, I should have, yeah, my bad. I actually do this personally all the time. I make that mistake. So this results grid view totally changes if you click like a peptide or a protein. So make sure you've clicked on one of these precursors. Yeah, sorry, that's my fault. I should have told you guys that. So I'll go through it again. So if everybody's got a particular precursor selected, so the thing right under a peptide sequence, yeah, hit, and you have the results grid open, you hit view, edit, or customize view, and then it'll bring this up. And now you know you're on the right track if at the very top it says precursor result. If it says that, then you can go down and find total area fragment, select it, and hit OK. And if it bothers you about a view name, just enter random characters. So the fragment, the area that it returns for total area fragment is already background subtracted for you. So when you say total area fragment, it's background subtracted area. Oh, yeah, this option here, total background fragment, will say if I sum up the background signal for every fragment, that's the number. And then if you hit OK and potentially type in a view name, it'll show you the data. Okay, so that's how to view it, just like in the results grid. But usually you would do this in the context of exporting a report. So I'll show you guys that too, and then we'll break for lunch. So usually if I was exporting a report for DIA data, what I would do is one like this. So if you hit File, Export Report, and then click on Edit List, and then select Add. Here's just an example of the type of report I would typically make. So if you choose from the replicates dropdown, you want to be able to see the replicate name. So in this case, I'll preview it. That's just the name of the file. And then typically you're going to want to know the name of the protein. So I'll select protein name up here. So now that's going to be a list of the replicates in every single protein. And then for the peptides, I would do the peptide modified sequence. So I went to get there. It's under proteins, peptides, peptide modified sequence. And then showing you that, that's now going to give you a list of all the peptide modified sequences. And if there's a structural variant in there, or structural modification, like this carbon monomethylcysteine, it'll show it in the sequence. Okay, and then finally, if you go to the precursors dropdown, you also will want to know, you'll want this stuff split out for different charge states of the precursor. So select precursor charge. And then finally, under precursor results, you'll want to expand that. And this is where it's key. Select total area MS1 and total area fragment. And then if you preview it, you can now see that this starts to become a useful spreadsheet to hand off to, I don't know, yourself. If you're a statistician, you can load it into R and do that kind of work with it. And you would generally want to be using this total area fragment column. However, it doesn't hurt to output the MS1 area. Potentially you want to try both. So now I can close this, and you'll need to name the report, so you can call it DIA report. Technically, in MS1, the total area is the same. Well, yeah, if you were only doing MS1 filtering, yeah. Yeah, so like the total area is going to take everything that's under that like filter tab, YBP, is going to sum up everything in there. So if all you had in there was P, then yeah, it's going to be the same. Okay. Oh yeah, sorry. Yeah, so this is the most important one. So to get to that total area fragment and total area MS1, I'll back out. It's under the subheading proteins, and then sublist peptides. Under peptides, there's a sublist precursors. And then under precursors, there's precursor results. And under precursor results, there's total area MS1 and total area fragment. Now that I've shown you the hard way to do it, here's the easy way. There's there's binoculars things. You can actually type it. So if you do like total area fragment, it'll find it for you. Or if you can't quite remember, just do total area. It'll jump around to anything with total area. I'm happy to say that was my feature request because, yeah. And then if you hit OK, you should have this report as an option. And of course, you can just hit OK and export it. So find the DIA report, hit export, and it'll give you a CSV file. And I will put that under Jarrett DIA tutorial reports, DIA report.csv, and there you have it. And then you give that to your statistician, and then you publish and sell. And that. And does anybody have any questions? So yes, this is a good question. So it's a little confusing, I guess. So the total area fragment is a background subtracted area. So it's like almost impossible to explain so that it'd be confusing. So like the total area values, the total area MS1, or total area fragment are background subtracted areas. And if you wanted to get like a not background subtracted area, you would take total area and add total background to it. Yeah. Yeah.
Is this only exportable to Excel?
I mean, it's a CSV, so like you can. It's called. Let me show you. So it's CSV is something that Excel can read. It's just a format. It's called comma separated value. So it's just like a text document, really. So if you actually open the file in something besides Excel, so like Notepad, for example, it's just a standard flat text file. So I mean, in R, it's like a one-liner to import this file. Similarly in Python. So this is something, if you're sending your data to anyone, like generally CSV, they'll know what to do with it. It's just like a very like interoperable format. Does that make sense? Is that what you were asking?
No, I'm just asking because sometimes if you have a lot of replicates and you don't do the thing,
Yeah.
it ends up that Excel won't.
Oh, because it's like over 2 million rows.
Yeah.
Oh. Yeah, so yeah, in that case, you should pivot it. Yeah, if you have a lot of replicates. Yeah, you could. Yeah, generally, I guess I don't. Yeah, that's a good point. I've never run into that issue myself because generally I don't use Excel at that level. Like, I think another thing you could consider doing is like filtering the report before you export it. So you can use, you're familiar with like the filtering stuff yeah. So you could try to filter it a bit. Or just like tell Microsoft that they need to make Excel work better with Skyline.